Hi, I want to give you an overview of the software called the spatiotemporal epidemiological modeler, or otherwise called STEM. Now, STEM, or the spatiotemporal epidemiological modeler, is a very interesting software that was researched and created by IBM Research and made available through the Eclipse Foundation. Now, it's a free open source software, and it is graphically based and can be used for a variety of simulations because it contains geographical visualization of the it contains geographical maps, and inputs and all that. So you can simulate and visualize the spread of diseases using this particular software. And let me quickly mention that this software is used specifically for infectious diseases. Now, this software contains a wide variety of mathematical models of infectious disease. So you have the compartmental models, such as SI, SIR, SEIR, okay? S for susceptible, I for infectious, um, E is for exposed, and R, of course, for recovered. So this software has a lot of information in it. It's an extensive set of predefined data in the form of graphs, transportation network, population data, weather data for each country. There are about 244 countries currently in this software. And scenarios that illustrate disease spread models around the earth can be done using this particular software. And the architecture of STEM is that it is Java based, and every STEM component is a plugin or bundle that can be independently developed, deployed, used, extended, and shared by various other researchers. Users can also create models for a country region and um built on existing models or create new ones. So STEM is quite extensible, flexible, and reusable. All right, so you can download this software from this website. Um, if you want to download and use, it is free. Okay, you can just go to this website and download it and then use it. It's a free software. Okay. Now, I want to talk about the STEM perspectives. In STEM, we have different perspectives. We have the designer perspective, in which you design your own scenario by putting in graphs of a particular country or different countries, and then adding some other components that you want in your scenario, and then you can run the scenario. Now we have another perspective called the simulation perspective. Now in the simulation perspective, the scenario you're running, you'll be able to observe it. In the simulation perspective, you'll be able to see disease spread across geographic locations. Then we also have the analysis perspective. In the analysis perspective, you can estimate model parameters from external data, and you can compare your various simulation results, and you can do some epidemic analysis. All right, now STEM has made it very easy for people to create and share their work. through what we call projects. And the project is a collection of files and folders. Okay, you can have projects developed by others. You can import them into your own STEM software and use them. You can also share projects, with other people, and it's quite easy to do. Okay, for the purpose of illustrating the way STEM works, you're free to look at some of these demo projects that are available for the STEM software. Okay, you can go to this particular website, okay, and download these projects, and then you can use them and see how STEM works. Now, I want to talk about design and simulation using the STEM software. Now, how do you create and run a STEM scenario? Okay, for details, you can go to this particular website that you're seeing on the screen. Okay, so in creating a scenario, you have a few steps. Okay, we can see these few steps graphically illustrated here. New STEM project, you need to click on the new STEM project, and then you need to uh, make a new scenario. Okay, you define a new scenario, and then new models. You need to define a new disease, define a new infector, and then you have your new sequencer, and then you'll be able to run your scenario. Now, in creating a scenario, it's important to remember that actually a scenario is an empty container, right? So without you putting things inside the scenario, you will not be able to run the scenario. Okay, so you need to add model containers for the model hierarchy, import graphs from the STEM framework, and you need to create the three nested models: the geography model, the population model, the disease model. You need to also create an infector and create a sequencer. Then you can equip the scenario with your disease model, infector, and sequencer, and then you can run the scenario. That's the minimum the scenario needs to run. Of course, later we'll see that there are other things you can also add to a scenario. Now, let's comment about the designer perspective. This is what it looks like, okay, graphically. You'll see that the designer perspective has been selected here. From this designer perspective, you can design scenarios, design experiments, whatever you really want to do, okay? Here we illustrate creating a new scenario, shift from the geography model that has the graph area, graph network, and all that. And then you can have your population model where you add the graphs of the population. And then you have the disease model where the particular disease you're working on is uh, added in the model. This is a three-level nested model. So you have the geography model, you have the population model, then you have the disease model. Well, what actually happens is that you take the geography model and add it to the population model, okay, and when you've constructed the population model, you add the population model to the disease model, okay, and then you're ready after defining the disease and all that to be able to add it to the scenario. Of course, you add the infector sequencer. Of course, you choose a finite, you choose a solver. Okay, choose a solver. Most of the time, people might use the finite difference, but other ones are also available. Okay, and then you can run the scenario. You also have multiple geographies, infectors, inoculators, and scenarios that you can run in STEM. Okay, you can have the geography of two countries or even more. Okay, for example, here this is Malaysia. Okay, so the geography of Malaysia and that of Thailand have been added together. They share a common boundary. Okay, so their geographies have been added together, and then infectors can be introduced, inoculators, and whatever you want in particular locations. In this case, an infector was introduced in a particular location in Malaysia, another one in a particular location in Thailand. All right, and then you can, of course, run the scenario and see what it looks like. And scenarios, you can run one or two, different scenarios. Okay, this is what the simulation perspective looks like. Okay, in the graphical STEM interface. This is the simulation perspective. You can see this particular simulation is running, and it can be visualized. All right, and of course you can control the simulation from here, and you can also observe various other things, under the simulation perspective. Okay, all right, so you can see the map of the particular places you are working on. You can visualize the time series, okay, using this STEM software. You can see under the time series, you can see the susceptible, you can see the infected, you can see the exposed, you can see the recovered. All right, so also you can output your data by clicking the CSV logger on. You can on the CSV logger, and then you'll be able to have your data generated for you, and automatically the data will be saved to where you saved your STEM installation software. Okay, you can run an automatic experiment. For details, you can go to this website, okay, to see the details about running an automated experiment. Okay, basically what you do in an automated experiment is you try to compose a scenario and simulate that scenario. And in an automated experiment, you're actually trying to perhaps find out unknown parameters from parameters that you know. For example, you have incidence data, and you want to estimate unknown parameters. Maybe you want to estimate a transmission rate, infectious mortality rate, stuff like that. You can use this automatic experiment to be able to do that. On the STEM software, on the menu, there is a provision for new automated experiment. You can select it, and then you can pick a particular folder that contains your incidence information in the format that STEM usually uses, the CSV format. Okay, and you can choose one of the scenarios found in your project. Okay, and then you can use this particular provision to try to tell STEM the unknown parameters that you want estimated. You can add them, the unknown parameters you want estimated. All right, so, and then you can choose an object that contains the parameters. You can choose a parameter, and then add them from existing projects where the scenarios you've already run. Okay, you can select that, and then you can run your experiment, and STEM will be able to estimate parameters that are unknown. Okay, you can get details and read further on automatic experiments from this particular website. Okay. In the analysis perspective, you'll be able to compare different scenarios. You can also do an epidemic analysis and look at an epidemic view. Okay, then there are built-in models, disease models in STEM. We have built-in disease models or multi-population, seasonal disease model. You have multi-population, SEIR model, experimental disease, SEIR disease model, several other models. Okay, they are already inbuilt in STEM, and you can use them. It is also possible to create a new model that is not existing in STEM, and creating a new model requires writing an Eclipse plugin. Okay, we have various population models in STEM. We have, for example, the aging population model. You can also create new population models in STEM software for your use. Okay, so you can have multi-populations in STEM software. Maybe you're working on a disease that affects animals as well as humans. For example, you might need to define a new species like birds, like pigs, mosquitoes. Okay, you can. We also have a provision for demographic population, like women, senior citizens. We also have aging groups, different age groups, right? And sometimes you have a disease outbreak modeling with mixed population and all that. There is provision for you to do some scenarios involving these different populations in STEM. But basically, what you do is to define a new population, okay, so that you can create a new population model, and you need to say what kind of population is it. In this case, in this example, for example, it's a bird population being defined, okay, and you can set other parameters when you are prompted to by the STEM software. And then when you've defined the new population, you will need to use this particular provision in STEM on the STEM menu. You need to use this new population initializer to initialize a new population that you've just defined. Okay, and then you add your new population model. You add your new population to the population model, and then you add the population initializer to the geography model. All right. Like I said, you also have aging population that you can use, okay, involving maybe different age groups. You need to define the new population model also for that and say what kind of population it is. In this case, for example, it is a human population model. You can also have the demographic population model, and this is the way to go about it, defining the demographic population model. Okay. We also have control graphs in STEM, and these control graphs, you can use them for a lot of purposes. For example, we have a control graph where you can be able to simulate the effects of vaccination and isolation of people as a result of an infection. Okay, there's a provision for it in the STEM menu for you to put a new graph, and you can choose what kind of graph you want. For example, control label graph generator. And when you create this new control graph, you'll be able to visualize the effect of like vaccination isolation, of what kind of control you're trying to do in that particular disease infection as it's spreading. Okay. Now, specifying population, fraction vaccinated, and all that. you can use the control graph and do this and specify the fraction vaccinated. For example, in this particular control graph, 0.3 fraction vaccinated, and then fraction isolated 0.1, and it's a controlled population for humans. Okay, all right. So you can do this, and then you can add it to the disease model. Specifically, you see your control graph is going to be among the graphs, but the particular designer perspective that you're working on, you'll be able to see under graphs, you'll be able to see a control graph. So all you need to do is to drag the graph and put it inside your disease model. And then you can run the scenario with that particular control graph, and then you see the effect of the particular control that you're doing on the disease. All right. And there is something we call a trigger, which we use in modeling interventions. Of course, as usual, you can see details on this particular website. All right, so sometimes you're running an experiment, the scenario, and all that, okay? And you've already defined your parameters and you're running them. Usually, those parameters you define are static, they don't change. Okay, so if you want to change them, what do you do? Okay, so one of the things you can do is to introduce triggers. Now, a trigger is actually an empty container. You will need to put a modifier inside the trigger and a predicate, and then you can add the trigger to your scenario and see what happens. Okay, for example, you can create a new modifier. And how do you create a new modifier? You right click on object and then check for create modifier. In this case, a disease is being modified, so the modifier is created, and you can indicate what kind of modification. Maybe you're modifying the transmission rate, the mortality rate, whatever. And then you would also, after you've selected the parameters you're modifying, okay, like is illustrated here, then you need to create a predicate. A predicate, it's a specification of when that particular modification you've made comes into effect. Okay, you want to specify when the modification you've made, when your modifier should begin to act. So in creating a new predicate, you need to select this particular in your menu. You need to select click on this button and select create a new predicate, and then you right click on your predicate, and there are a lot of options. Okay, a lot of options. In this case, we'll be using this option of elapsed time test. We want to say maybe after so number of days, our modifier begins to work. In this particular example, you're seeing elapsed time is 20 days. So modifier will be activated after 20 days. You see the effect of the modification you've made of maybe reducing or increasing the transmission rate or the incidence or whatever modifier effect you want to see in operation. Okay, of course, there are other provisions, like I've said, in the predicate that you can use, and you can get some more details on how you trigger interventions using advanced predicates. Okay, so when you've created your modifiers will be located in modifiers on your STEM perspective, designer perspective. You see on the extreme right panel, under modifiers, you see all the modifiers you've created. They will be there. The predicates you've created also will be under provision for predicates. All right. So, and then You can use them in different scenarios by adding the trigger to a particular scenario. You'll be able to see the effect of your modifier and your predicate, which are contained in your trigger, added to a scenario. Okay. Experiments can also do experiments. These experiments, you run them in batch mode. Let me quickly say that this particular experiment is different from the automatic experiment. Okay, so simulation usually executes a single scenario, but sometimes we need to run experiments, so different runs of the same experiment, the same scenario, but with different transmission rates. In order to be able to do this, we use experiments to run scenarios in a batch mode. Okay, now you need to switch on the logger to log the data as CSV files that you want to use. All right, so in experiment, you have your scenario, and then you have a particular modifier effect that you want to observe. Okay, and then that modifier effect is added to your scenario, and then you run the scenario. All right, so to create a modifier, all you need to do is go to the provision for modifier on your STEM software, okay, and then do the particular modification you want, maybe your transmission rate, like we've seen earlier under interventions, right? But in this particular case, you're going to be using a range, okay? You can say starting value is maybe 0.8, for example, for transmission, and maybe end value is 1.2, but you want this range to be moving at a step of 0.1 until it gets to the 1.2, right? So you can define it here, 0.8 to 1.2 by 0.1, okay? You want it to be moving by a step of 0.1, okay? And you want to be able to see in different batch modes what the effect will be like, okay, of this different change of a particular scenario, but different batch modes, as it were. All right, so for you to do a new experiment, you need to come here, select this new experiment, and then you will need to add your scenario plus modifier to the experiment. All right. Of course, not a slight difference here that in triggers, modifier is added to trigger and trigger is added to scenario. But in this case, you just add your modifier straight to the scenario, right? Under your experiment, and then you're ready to run the experiment, right? So you can read more about what STEM can do for you. You can watch our YouTube videos. We have different videos, okay? We have a lot of websites where you can get a lot of stuff about what STEM can do for you, okay? There is so much available online. I want to thank you for listening to this brief overview of STEM. You can actually contact us through the emails that are on the slide that you're seeing. All right, so if you have some questions or some clarifications you need on the presentation, please feel free to contact us. Thank you very much.